So good afternoon, everybody. I think this is on now. Jonathan Yingling, I'm the CSO at Arcus, and very much appreciate the invite from the organizers, and perhaps even more so the flexibility to allow me to go first so that I can catch a flight home this afternoon. So, and I also appreciate Tony's introduction and the talk that he gave a little while ago on the activity of casdatifan in the Cas-Cabo combination in particular. And what I'm going to cover today will look redundant initially, but I promise you I will get to the biomarker and translational strategy that we are deploying within our casdatifan program with some early highlights of potential associations with outcomes. This audience knows well the transcriptional profile that HIF2 alpha and HIF1 beta modulate across a whole variety of different pathways that are involved in tumorigenesis, and the mechanism of action for casdatifan, very similar to belzutifan, is to block that interaction and thereby disrupt the transcriptional program that leads to tumor progression. The studies that we have highlighted so far, ARC-20 is the phase 1 dose escalation as well as the dose expansion study. What I will focus on today are those 3 cohorts in the middle, the second line RCC at 50 milligram BID of casdatifan, 50 milligram QD of casdatifan, and then the more recent 100 milligram, which, as Tony highlighted, is the dose being carried forward into phase 3. There are additional expansion cohorts that I will not cover today that Tony also highlighted earlier that are in progress. Very quick update. So 2 data sets from the ongoing ARC-20 studies. The update here is that we do now have the first estimate of the median PFS in the CAS mono second line cohort. This is the combined 50 milligram QD, 50 milligram BID cohorts across with a 5 to 15 month follow-up. The response rates are in the range of 25 to 33% per cohort, as Tony highlighted earlier. Importantly, the rate of primary progression is less than 20%, and the median PFS in this ongoing, still immature data is 9.7 to 12 months plus. So several of the tables that we saw earlier today where the casdatifan monotherapy PFS, the ORR was there, the median PFS was blank in those tables. Please update with the 9.7 to 12 month current estimate. And that'll be updated as the study continues to mature. What I really want to focus on today is the clinical biomarker strategy that we've deployed within ARC-20 and that we will also carry forward into the PEAK-1 trial. 2 major components, the longitudinal serum and plasma samples, as well as you'll see in a few minutes, some pre- and post-treatment biopsy samples that we have had access to. In the context of collaboration and requests within this community, as you will see, we have exactly 2 pre- and post-treatment samples from within ARC-20 where we can begin to draw some conclusions, but it is literally n equals 2. Understanding that it's not standard of care, it's not standard practice to have on-treatment biopsies. They're invaluable samples as we learn about how casdatifan is working and resistance mechanisms into the future. And to the extent that it's possible within the context of these trials to collect those samples, they are tremendously valuable resources as we try to benefit patients into the future. In addition to the longitudinal serum samples that I will talk the majority about today, we do have HIF1 and HIF2 alpha immunohistochemistry that we spent time internally validating to a very high standard, where we will be evaluating genomic alterations, VHL and others, by whole exome sequencing, and we have transcriptomic analysis that we will be analyzing by RNA-seq as well. What I will focus on is what's shown here on the right. So EPO, we all know EPO is a pharmacodynamic marker for this mechanism of action in the periphery. One of the things that we noticed as we evaluated our ARC-14, which was the initial healthy volunteer study for casdatifan, relative to the levels in our ARC-20 RCC patients, there's an elevated level of EPO across those 2 populations. Shown in green is the normal level of EPO, and as you can see in RCC, there tends to be a higher level of EPO in the periphery. Just a reminder from the clinical data in the casdatifan ARC-20, both the dose escalation as well as the data that we have in the expansion cohorts, the level of casdatifan that is giving equivalent EPO modulation in the periphery to the approved dose of belzutifan at 120 milligrams, That gives around 63-64% inhibition by belzutifan. The dose of casdatifan that is modeled to predict or modeled to deliver the same level of EPO modulation in the periphery is 20 milligrams. And as a reminder, our dose into phase 3 is 100 milligrams moving forward. So we like to think of this as 5 times the equivalent dose of belzutifan that we will be evaluating as a HIF-2 alpha inhibitor in RCC patients. You also saw earlier from Tony that the profile of the response to EPO is distinct between what casdatifan can deliver in terms of a prolonged mediation of EPO suppression compared to what's published for belzutifan, where you get this rebound effect over the period of 13 weeks. Importantly, we have been able to analyze in those 2 pretreatment biopsy samples the EPO response at the mRNA level in response to casdatifan. And you can see a reduction in the tumor for the EPO mRNA, as well as 2 additional HIF-2 alpha target genes, very well known, PTHLH and SERPINE1, both being reduced in these pre and post-treatment biopsies in response to casdatifan at 100 milligrams. We've also begun to analyze in terms of associating the response of casdatifan, the best EPO response, in relation to progressive disease, stable disease, and CR and PR. And what you can see is there's a relationship between the best EPO change delivered with casdatifan from PD to SD to CR and PR, where there's a statistically significant change, further reduction in CR and PR compared to PD, and between SD and PD. Likewise, we've looked at the profile, dividing at the median for EPO response to casdatifan into 2 populations, what we refer to as a deep response, below the median of 85% casdatifan reduction of EPO, or above the median, shown on the right, and there's an association of the response rate in the deeper response to casdatifan at 37.5% compared to the 17.5% for the less deep, and the progressive disease is also lower in the deeper responding patient population. And then finally, as Tony highlighted earlier, and I won't belabor this, we're on the verge of initiating both the PEAK-1 study, 100 milligrams in the post-IO setting in phase 3, as well as the EVOLVE study in collaboration with our AstraZeneca colleagues. Thank you very much.
So now we have time for questions. Hans has a question down front. Do we have folks and Jim in the back?
No, I'm in charge.
How did you get in charge of this? What could go wrong?
That's not how it does. And that's how it works. So beautiful work, obviously, very interesting agent, very promising agent. But you also heard some concerns about, HIF-2 inhibition interfering with immunogenicity of the tumor, et cetera. You have a lot of samples, so to say, plasma samples. Are you looking at inflammatory markers, changes with the treatment of HIF-2 inhibitors? Same with the pre and on treatment biopsies. Could be very invaluable to look at the tumor immune microenvironment.
So the answer to both of those is yes. Unfortunately, in the pre and post treatment biopsy tissue samples, we really only have those 2 samples, so it will be very difficult to draw conclusions from an n=2. And it really is a request that when it's medically feasible and safe for the patient on casdatifan to be able to achieve a post-treatment biopsy, that would be a very valuable resource for us. In the periphery, we are doing additional analysis, cytokine analysis, as you can imagine. And at baseline, we are also doing very broad transcriptomic profiling, signatures, et cetera, associated with overall response rate for now. And we very much look forward to looking at it in terms of PFS as well.
So we're going to ask one more question on this session, which is David, and then Kura's going to present.
Okay, so John, you showed the EPO data. Is either your data or the Merck data potentially impacted by exogenous EPO? Do you know that?
Yeah, so it's a great question. So if I can just go back.
Probably.
Not going to work. So yes, it is. However,
yep.
When we measure EPO, a patient that is administered an ESA is an excursion to the up to the very high levels of erythropoietin, as you can imagine, and those are censored and removed from this. So the profiles that you see account for and remove the very high excursions that you see.
For both yours and the Merck data, or just yours?
I can't speak to the Merck data.
Okay, that makes sense. Tom, are you still taking questions?
Tony, is it really important? Is it really important?
No. Okay. No, I just want to say, I think you've done preclinical work, I mean, to Hans' point, not in clinical samples where you expose you expose cell other to CAS, and there was no major change in the tumor microenvironment or decrease in CD8 T cell, et cetera, which was kind of reassuring.
There's no negative impact on the combination with PD-1.
It's not presented here.
Correct. So this is all the clinical data. We are looking in the post-treatment biopsy samples with a caution that we literally have 2, and we're looking in the periphery for any impact on the immune system.
Okay, everyone, thank Jonathan. Good luck with your flight.
And my apologies that I have to run, but I very much appreciate it.
All right, is it Jabed, are you coming next?
No, Jabad has the next flight to leave, so Terminal 2 in 45 minutes. I'm sorry. Pleasure.
Thank you, KRS, for the opportunity. And the first day, the very first session, if you had a chance to attend, actually, let me see which one is forward here. This one?
Actually, the green one.
The green one. Oh, green one? Okay. So in that, before, does it go back? No.
It does.
Okay. Anyway, so my disclaimer, I'm actually a clinician, I'm a medical oncologist, and a full-time employee of Kura. And going back to this very first day, very first session of the first day, wonderful speakers and our wonderful moderator, Rana McKay, actually did 2/3 of my job, which you can see here, the first 2 bullet points. So it's established, let me directly jump into the last bullet point here, which is what is KO-2806. This is a second generation or next generation Farnesyl transferase inhibitor that blocks the hyperactivity growth factor signaling by blocking mTORC1 by inhibiting farnesylization, which is required to persistent activity for mTORC1. And so that's why we had chosen to combine cabozantinib plus this in this RCC model. I'm going to share with you a few preclinical data in various mouse models. This is a TKI, as you can see, this is a TKI naive RCC models. As you can see here, that you have a very good response, cabozantinib, but if you add KO-2806, further in-depth response you can see, or further deeper response you can see. On your right side, that's actually, you could see that's a quantitation, that adding 2806, you see the depth of response. And the next, as you can see in IC, it's not only anti-tumor effect, but also we have seen an anti-angiogenic activity of cabozantinib in RCC as xenografts. As you can see, cabozantinib in the ISC model, but if you add 2806, it's basically, have a further in-depth response of anti-angiogenic activity of cabozantinib. And on your right side, you can see the quantitation. And if you add both cabozantinib and as well as 2806 individually have shown the activity. However, when you combine with 2806 and cabozantinib, you see a statistically significant difference over there. And then this is a more model, xenograft models, as you can see, RCC tumor progressing on anti-VEGF or TKIs. Here, 2 examples. Lenva is there. In Lenva plus Lenva is working, and then afterward it has started to grow. And then as you add with this cabozantinib, it produces a better effect. However, when you add 2806, it's produce a deeper response on that. And same thing on your right, you see this. So based on these data, we have encouraged to start our first-in-man study called FIT-001. As you can see, the clinicaltrial.gov is over there. The trial is very active at this moment, and it's basically 3 different stages of the trial. First stage, obviously, to ensure as a monotherapy, the product is safe and tolerable. Then we have second intention was to determine the MTD, and subsequently, once we had settled the MTD, we started to combine with other agents at the second stage. Again, main objective was to prove that the product, the combination is safe and tolerable, and in addition, if any anti-tumor activity. So we are at that stage at this moment, almost near finish, completed to the combination dose escalation stage. And the very last stage would be once we have tested in each combination about 6 doses, as you can see, then we'll have option to choose 2 doses to take it further in expansion in a randomized fashion, or if there is a clean winner within that 6 doses we have tried, that would be actually, we could take it as a lone cohort further for the expansion stage. I encourage to go to this nct.gov and then see this clinicaltrial.gov and see this further details of the trial. And unfortunately, I couldn't share with you data for ESMO embargo. We just had these things.
Very sad. Very sad.
Disappointing.
Yeah, there are multiple abstracts has been accepted in ESMO. Just wanted to let you know. And it's basically open the study. As I said, we are in the middle of the study. We have 16 sites in US and few sites in Europe, several countries. And as you can see, the very last bullet point, first disclosure would be at ESMO this year. And finally, I would like to acknowledge the patients and their families and their caregivers, our investigators, whom actually I have seen few of them yesterday and today around here. And the study is obviously sponsored by this company. Thank you very much.
Magic.
Thank you very much.
Are there any questions from the audience?
Any of the investigators want us to talk about the trial?
I've got a question here. Darren, I think you've got a question.
Just had a question about whether there was a biologic rationale for combining with cabozantinib versus other VEGF receptor TKIs.
So, yeah, so we discussed internally, but at this moment for this is our own asset, this second generation or next generation Farnesyl transfer inhibitor, and it's a, company have decided to pursue that path, but yes, obviously we continuously discuss with other combinations, yes. In this current study, you would see actually there are other combinations, other drugs. I focused here RCC, however, we are also, it's actually public domain. We are also pursuing non-small cell lung cancer, colon cancer, including pancreatic adenoma ductal cancer. So that's actually in public domain. You can see it.
Okay, great. Sounds good. Have a safe flight.
Thank you.
Thanks
Best
for
of
coming.
luck. Thank you very much indeed.
Thank you very much. We look forward to future updates. The next up is Hans Hammer's favorite agent. Randi Isaacs is going to talk about...
That's not me. I'm not the agent.
Right. Right. Randy's going to talk about a new masked form of IL-2. Sounds very exciting.
Yeah, green is forward. Great.
So I thank you to the organizers for inviting Werewolf Therapeutics to present our masked, our conditionally activated WTX-124 IL-2. We've also been allowed to use our own slide format, which I thank them. Werewolf was formed in 2017 with the express purpose of taking agents that were known to be active, but were particularly toxic and had very poor therapeutic index, and then increasing that therapeutic index. We specifically started with cytokines, and INDUKINE being inducible cytokine. The idea about Werewolf comes from the fact that we are benign in the periphery, but when we get into the tumor microenvironment, we actually become dangerous agents. And so that was the rationale for the name of the company. The challenge, I'm sure, as everyone in this room knows, with high-dose IL-2 is that it does have tremendous toxicity and is really limited in its use, although it was the first agent that actually produced durable responses in patients. Unique advantages. Is that true?
Yes, you're among friends here.
I know, okay.
You're not supposed to be interacting with the panel.
I'm sorry.
No.
I'm very supportive. She looked at me like I was going to say
I can
something.
see it. I can see that.
So the unique advantage of WTX-124 is this is a novel prodrug. It was engineered with enhanced pharmaceutical properties to only be active in the tumor microenvironment. A number of key opportunities here, as you must know. This is a picture of WTX-124. It is a tunable, tissue-targeted therapeutic. We now are saying cancer and other diseases because we actually have a molecule that can be used for inflammatory bowel disease. But I'm specifically going to talk about the 4 sections of the molecule. And this is a single polypeptide. So most importantly, we use a wild-type cytokine. So we are not skewing to any receptor, we're not mutating, and that's critically important because native IL-2 has activity. We have a blocking domain, and this is specifically tailored to the molecule, prevents activity in the periphery, and that's what confers the therapeutic index. We also have an anti-HSA domain, which increases the half-life and allows for increased exposure over time for the molecule. And then those little red pieces that you see poking out there are sort of the secret sauce. Those are agnostically chosen sequences that are cleaved by proteases within the tumor microenvironment. And as I'm sure you all know, there is a number of proteases that are dysregulated in cancer. We specifically chose sequences that were chosen by a library of human tumors that were dissociated. So for any particular patient, we actually don't know which protease cleaves this particular tumor, but we know we cleave across a broad range of tumor types, and actually we cleave best in renal cell carcinoma. In addition, we don't cleave in normal healthy tissue. So this is a little schematic, is how the INDUKINE works. You can see the protease dissociating, and then the cytokine binds to its receptor. Right. Specifically want to show you in the top panels, and this is some preclinical data. All of our preclinical data has been reported. If you look at the plasma versus the tumor, the light blue is the prodrug, and the purple is the free IL-2. And you can see in the plasma that you have a large amount of the prodrug and very little of the free material. But you go to the tumor and you see that it's almost reversed. You still have prodrug that's completely bathing the tumor during the dosing period, and for our clinical applications, it's every 2 weeks. But you see in the tumor also that you now have a large amount of the free IL-2 that is continuously available to the tumor. So we are in phase 1b. We actually are fairly far along. Patients enrolled in this study were those who had immunotherapy-sensitive tumor types, so any tumor type for which a checkpoint inhibitor is approved. We had monotherapy dose escalation. We also had combination therapy dose escalation with pembrolizumab. We went through a number of different cohorts. We started to see clinical activity at 12 milligrams delivered IV every 2 weeks. And at 18 milligrams, we expanded our patients and decided that that was the recommended dose to move forward with. both for monotherapy and for combination. Very important to note that we did not see any increased toxicity with the combination, and thus were able to use the same dose for both mono and combo. We have several expansion arms that are all open. We have renal cell carcinoma for both monotherapy and combination. We have cutaneous melanoma for both monotherapy and combination. Cutaneous squamous cell carcinoma, I'm going to show you a particular patient who had a remarkable response. And we also have non-small cell lung cancer in PD-L1 positive tumors. I do want to say that we have a very robust biomarker plan that I'm not going to be able to speak about, but we do get pre and post tumor biopsies on virtually all of our patients, and that started with the very first dose of drug. So this happens to be a metastatic cutaneous squamous cell carcinoma patient. I wish it were a renal cell, but at this point we are just getting our renal cell data in. But it's very interesting because it shows the power of IL-2 and the power of WTX-124. So this was a man who had significant disease in the premaxillary area with extension back into the pterygopalatine fossa. And the patient had was primary resistant to the standard of care checkpoint inhibitor, which is cemiplimab. Patient initiated treatment with 12 milligrams IV every 2 weeks of WTX-124. And the baseline CTs and PET scans you can see there, at 3 weeks, and this is the property of the drug, you know very early whether the patient is going to respond. An on-treatment biopsy showed no tumor. And in fact, the investigational radiologist, interventional radiologist went in and said, I can't find anything. And at 8 weeks, we had a partial response. At 12 weeks, PET scan was negative. The patient stopped treatment at 21 weeks in the setting of a confirmed complete response. And this was because we are a microenvironment, tumor microenvironment activated agent. And so without a tumor microenvironment, it made no sense to continue drug. The response is now ongoing at greater than a year. So other data from the trial, safety, I'm sure everyone is curious about. Despite the fact that we give greater than 120-fold more drug than has been given before, we see no evidence of vascular leak syndrome or cytokine release syndrome or infusion reactions. Majority of the events were grade 1 and 2, and we have no new safety signals in combination with the checkpoint inhibitor. From a clinical activity standpoint, we've seen confirmed durable responses both in monotherapy and combination therapy, and we've also seen from a biomarker standpoint dose-dependent increases in activation of effector T cells in the tumor microenvironment, and this is by IHC-IF as well as nanostring. So I hope for next year to be able to be invited back and have renal cell data to show you. As I said, we're actively enrolling in all of our expansion arms, and we're very, very excited with the data that's coming out at the end of this year. Thank you.
So do we have questions for Randi? I'll run over with the microphone, or Jim in the back.
Jim in the back there.
Let's,
Got to go a bit faster, David,
sorry,
otherwise
sorry.
this doesn't work if you walk.
Thank you. So I couldn't see your graphs very well from the back, but it seemed it was a log scale. It seemed that the scale that you were showing for concentrations was a log scale, yes. In which case it seems that the amount of mature cytokine is probably 1/1000 or so of the plasma levels. Is that correct?
In the tumor. You're talking about the amount in the tumor versus the periphery?
Compared to the plasma.
Yes, you are correct.
So even though you're 120, giving 120-fold more drug than with high-dose IL-2, you're still giving 10-fold less.
Well, I'm not sure that anyone ever measured how much IL-2 is actually in the tumor with the high-dose IL-2. We know that we get enough, and remember, with high-dose IL-2, you have a very short half-life. And so what we're seeing is this continuous bathing of the tumor by the prodrug and continuous cleavage. And so there's an extended period of time that the tumor sees the WTX-124, or actually the active IL-2.
So how did you determine that you were 120-fold more drug?
So I didn't show the PK curve. It's actually the Cmax. It's actually the Cmax. So if you get the Cmax of high-dose IL-2, we are 120-fold less of free IL-2 than the Cmax of high-dose IL-2.
Okay. So my last question, have you tried, so you show one model, have you tried this in PDX models of renal cell carcinoma? How translatable is this from model to model? You mentioned that your linkers are susceptible to different proteases. So, and I imagine there's going to be substantial variability across tumor types. So do you have a sense for how representative the model is and how many kidney cancer models have you tried?
Yeah, so that's very interesting. So as you know, with IO, we have to use syngeneic mouse models. We can't use specific models for different tumor types. And so the working horse model for IO is the MC38, and it happens to be a colorectal cancer model, but it is not necessarily, reflective of, it's more for how does this work versus it's specific for a particular tumor type. So there isn't a syngeneic model that is specific for kidney cancer. And we can't use, xenograft or other models with these molecules.
Okay, I think Laurence has a question.
Yeah, quick question. What will be your combination strategy?
Well, right now we are combining with pembrolizumab, but especially, we think about how many different tumor types one can treat with IL-2. The potential for combinations is enormous. And we're also thinking about, other lines of therapy, second line of therapy. Monotherapy is an option for this drug. There are patients who can't receive a checkpoint inhibitor again. And then with renal cell, you have the ability to combine with a number of different agents.
That's magic. Thank you very much, Randy. We're now going to ask Robert Awadala, who's going to come and talk to us about CA9 radiopharmaceuticals in RCC. Robert, welcome.
So I'm very happy to be here. Thank you to Dr. Powles and KCRS for having me. My name is Rob Awadala. I work within the Medical Affairs Department as a medical science liaison for ITM USA. My talk today is on ITM's investigational CA9 targeted radiopharmaceuticals for both the diagnosis and treatment of patients with CCRCC. This is our phase 1, phase 2 trial design and first-in-human CA9 images. So the most important part, or the most important statement on this slide, is that 85 to 90% of CCRCC expresses CA9 due to HIF activation as a result of VHL tumor suppressor loss of functionality. So when the von Hippel-Lindau gene is mutated, the issue here is that it causes overactivation of HIF-1 alpha, which then in turn causes the overexpression of CA9. So this makes CA9 a very attractive target. I know a lot of the talks today were about blocking, HIF, but with regards to CCRCC, we're focusing on CA9, a very attractive target. So for our radiopharmaceuticals, we have ITM-94, which is a peptide bound to gallium-68, which is a PET radionuclide for imaging purposes of CCRCC. And for our therapeutic, we have ITM-91, and it's the same peptide bound to lutetium-177, which is a beta-emitting therapeutic radionuclide. So both ITM-94 and 91 form a true theranostic pair because they are using the same targeting peptide. This is our phase 1, phase 2 trial design. You'll notice some unique features here that it does include our diagnostic agent as well as our therapeutic agent. I'll briefly go through it. Part A is our phase 1 imaging for ITM-94, which again is our gallium diagnostic radiopharmaceutical, and that's being evaluated for safety and tolerability. Part B is our phase 1 trial, and in that particular part of the trial, patients will receive ITM-94, which is our gallium diagnostic radiopharmaceutical, followed by ITM-91, which again is our lutetium therapeutic radiopharmaceutical. And you'll see the dose 1, dose 2, dose 3, and that is because it is a dose escalation trial to determine the maximum tolerated dose. When we arrive at the recommended phase 2 dose, that will then start part C of the study, where the lutetium will be evaluated for its anti-tumor activity. Part D of the trial is kind of an add-on, and Part D of the trial is ITM-94 again. That is our gallium diagnostic radiopharmaceutical, and it's being evaluated for indeterminate renal mass. And of these trials, Part A, the enrollment is currently completed. Let's look at our phase 1 first-in-human ITM-94 imaging results. You'll see from the images here that ITM-94 has tumor-specific uptake, and it was observed 15 minutes to 4 hours post-administration. Additional ITM-94 imaging results. In this slide, you see that 3 patients were dosed with ITM-94, which again is our gallium diagnostic radiopharmaceutical, and it was determined that 1 hour plus or minus 45 minutes post-ITM-94 administration was the optimal tumor visualization time, and 5 millicuries, or 185 MBQs, was the optimal dose. This slide illustrates, and it's illustrated in the blue box here, that ITM-94, which again is our gallium diagnostic radiopharmaceutical, identified 17 lesions not detectable with prior conventional CT. This is our ITM-91 therapeutic dose escalation. So if you recall, patients receive ITM-94 and then ITM-91. So on the left-hand side of the image here, patient received ITM-94, which is our gallium diagnostic radiopharmaceutical. You could see from the images there, well, the image there, that in addition to CCRCC, the patient has a large number of metastatic lesions. Well, after 3 doses of ITM-91, shown on the right-hand side here on treatment, the patient has less lesions. So what are our take-home messages? Due to high CA9 expression, 85 to 90%, CCRCC is a promising target for both ITM-94 and 91. As reported by Dr. Hoffman in his published paper on ITM-94, which is, again, our gallium diagnostic radiopharmaceutical, as part of Part A of the ongoing Phase 1, Phase 2 trial, it was reported no clinically significant toxicity findings, rapid and sustained lesion-specific uptake, detection of more tumor lesions than standard CT, and tumor visualization results were available 1 hour post-administration. ITM-CA9 Phase 1, Phase 2 trial is ongoing. ITM-94, which again is our gallium diagnostic radiopharmaceutical, is being investigated for indeterminate renal mass. This is in addition to CCRCC. And ITM-91 is undergoing therapeutic dose escalation and is enrolling patients with advanced CCRCC. Thank you so much.
I think really interesting, Robert.
Questions, Raina?
We've got a question. Yes, Raina, far away. Yeah.
Is that okay? Very nice presentation. I couldn't help but notice the significant uptake in the GI tract and the images that you saw. So are you, I know you're just starting to treat patients, like what is the anticipated toxicity profile for this agent, just given the uptake throughout the gastrointestinal system?
You raise a good point. There is CA9 expression in the GI tract, and that's why you are seeing that. But I'm not able to disclose any information about anything further than that.
Tony?
What about disclosing, what about the liver? How's the LFTs on these patients? Did they go up? Because, you know, CA9 is expressed in the small biliary ducts. you can't disclose, even for me.
Right. Thus far, it's very good because remember it's a peptide.
Correct.
We with regards to peptides, they're usually renally eliminated, but at this time and point, that's really all that we know.
Yeah. Oh, that's good.
I've got a question. Is there a question? I've got a question, if I may. In terms of the imaging, what is the heterogeneity and the SUV max? Is there a huge amount of variability between metastasis? And is there a correlation between intensity of uptake and response?
Right now, we're still in the imaging portion. The results have not been analyzed to actually answer that question. It is a fabulous question, though. But what I will say is that we're seeing it for CCRCC, and as you saw from the slides, with regards to metastasis as well, because what's happening is it binds itself to CA9. So whether CA9 is just in the kidneys or whether it's metastasized, the radiopharmaceuticals tend to bind to it regardless.
We saw maybe data 2 or 3 years ago at ASCO GU, and I don't feel we've moved significantly forward from we haven't announced big randomized phase 3 studies. As if it was a VEGF TKI, we probably would had big trials and thousands of patients. Why is this field not moving as quickly as some might expect or hope?
That's an excellent question. I could speak from our perspective. ITM did purchase, this public information, the study from Debiopharm. So I can't speak for what they were doing with the study. I know they were a smaller company. But beyond that, I know there's some other companies targeting CA9. I think it's wonderful that everybody's talking about HIF and HIF inhibition, but we're a little bit more downstream, where we're going to let HIF do its job, which unfortunately is not a good job, but we're coming to bind CA9. But I can't speak further than that. I really think it has to do with the fact that it's the disease numbers, the population is not as large.
Where would you like radiopharmaceuticals to be 2 years from now? Would you expect them to be in phase 3 trials, in phase 2 trials? Where do you expect the field to be with a tailwind, bit of luck and a bit of money? Where would you expect the field to be in 2 years' time?
I definitely expect it to be in phase 3 trials because if you notice, our trial is diagnostic and therapeutic, which means that it would probably take longer to market. But however, when it does get approved, both of the diagnostic, the entire theranostic pair will be available. But I definitely see within 2 years it being in phase 3.
Susan, I apologize. I can see you were there for a while. That was slightly rude. I'm terribly sorry. It's
out.
poor chairing, to be honest. Yeah.
I noticed on the imaging slide that you said that you got more lesions recognized with the lutetium agent than the gallium agent. Were there any examples of lesions which were missed with the lutetium but appeared on the gallium scan?
So a couple things to clarify. ITM-94, the gallium is just the diagnostic portion. It's there just to identify, see the images of the lesions. And ITM-91, the lutetium-based, is there to treat it. So on the slide that I was showing you, it was just illustrating that the lutetium acted as a therapeutic agent to reduce the number of metastatic lesions. So it's not a matter of the lutetium showing more or less lesions. It's a matter of the lutetium treating the lesions and the patient having less lesions after being treated with the lutetium portion. The gallium portion was just to identify. Does that answer?
Are you happy, Susan?
Yeah, I wondered if there were any tissue-specific differences.
So the good news is we're 3 minutes ahead of time for those of you who are sticklers for time, and that's partly because of David's chairing. And it allows us to move to Jonathan Powles from Calico Labs, who's going to talk about a novel therapeutic approach to treating cancer, which is exciting.
Thanks. So I'm Jonathan Powell. I'm the head of drug development at Calico Labs. So immunotherapy cures cancer. Yet in spite of that, immunotherapy is out of favor. So I'm really excited to tell you about our novel drug, which hopefully will you'll be just as excited about. So ABV-CLS-484 is a PTPN2 inhibitor. PTPN2 is a phosphatase which negatively regulates signaling. And the really neat thing about this target is it both enhances immune activation, for example, T cells, NK cells, myeloid cells, but also it targets the tumor. So this is where it's different from your typical checkpoint blockade. It causes increased response to gamma interferon within the tumor, making tumor cells more susceptible to immune-mediated killing. In terms of the preclinical data, it's actually very robust. Quite frankly, I would say, if not equal, better than what we see in the lab in terms of PD-1 and C2A4. And so what I'm showing you here is the perfunctory MC38, where it wipes out the tumor. 4T1, where PD-1 doesn't do very well, we do a little bit better. CT26, a combination with PD-1. And then also relevant to this group, we've done a number of experiments with VEGF TKIs, in this case, Cabo, we get good responses. And then also, it has great effects in terms of enhancing cellular therapy, and for multiple reasons. So for one, it enhances homeostatic proliferation. So after you adoptively transfer T cells or CAR T cells, it causes an increase in the number of cells within the body. But then also it promotes persistence, and that's a key element of the success of cellular therapy. And then finally, one of the biggest problems in CAR T cells has been the beautiful efficacy in hematologic malignancies and the not so good efficacy in solid tumors. Now, this has been attributed in part to the fact that solid tumors need interferon gamma signaling in order to be susceptible to CAR T cell killing. Well, I told you one of the mechanisms of action is enhancing responsiveness to interferon gamma at the level of intrinsically in the tumor. So multiple different mechanisms by which this drug can help to enhance immunotherapy. So currently we're in a phase 1 trial, and for antibody-mediated therapeutics, it's great. You just give it every 4 weeks or so. But really, there's not been a small molecule inhibitor IO therapy. And so it's been taking us some time in our phase 1 trials to figure out the proper dosing as well as the regimen in order to know how best to deploy this. But in the meantime, as we've been doing this in all comers, we're starting to see evidence of activity. And one of the things that we've seen in a number of patients, which I want to highlight here, is that there have been a number of patients that are currently on anti-PD-1. So this particular patient I'm showing you with the gratuitous CT scan is they were on PD-1. In December, their physician said, you're just blowing through it, we're going to take you off and enroll you in a clinical trial. They joined our trial in February, and then they started to respond to our agent, indicating that there's not necessarily the same mechanisms of resistance. Again, this isn't a patient that was resistant to PD-1 5 years ago and then was treated with all sorts of different things and then is coming back to immunotherapy, but really in the immediate resistance time frame. So we're super excited about deploying 484 in renal cell, and mainly because, again, in our all-comer phase 1 trial, we've seen really nice, if you will, evidence of activity in the renal cell patients. A number of the investigators here have had good response in their patient. You can talk to them about it. And so as a consequence, what we're going to do is, beginning in August, we're going to convert our all-comer phase 1 to expansions in renal cell carcinoma with 2 combo doses and then a monotherapy dose. And our plan is, if we see the activity that we're hoping to see based on the smattering of experiences we're having in the all-comers phase 1 part of the study, then we're really going to gun for a registrational trial in renal cell. And certainly we look to all of you to help us decide where there's an unmet need and where we could be most impactful. So in summary, ABV CLS-484 is a novel immunotherapeutic drug. It targets both the tumor and the immune system. We have really robust preclinical data for combinations with checkpoint inhibitors. And I'd like to add is that we've done a lot of, in our phase 1, there's been a lot of combination, and you don't see the toxicity that you see with the ipi-nivo when we combine pembro in our drug. And so that's really a potential opening to treat renal cell patients with 2 immunotherapeutic agents, but maybe decrease the toxicity. Likewise, if you really believe in immunotherapy as the cure for disease, there's the opportunity to do triple therapy, particularly since this is a distinct mechanism of action. Again, we've had some nice preclinical data with the VEGF receptor TKIs, looking to open up a trial to try it in combination with TKIs in patients. And then finally, during this meeting and certainly at other more general immunotherapy meetings, it's clear that cellular therapy in the form of CAR T-cell therapy or adoptive T-cell therapy, again, is able to produce these durable remissions. And so if you have a trial or if you have a cellular therapy, which you're getting good responses in renal cell based on the multiple mechanisms of actions, we think that if you add the P2N2 inhibitor, you can only make those results better. So thank you very much.
Excellent talk. Looks like we have a few questions for Jonathan. Laurence, you want to go first? You have the microphone.
Thank you very much. Great presentation. So based on the mechanism of action, do you foresee this more as a rescue strategy in PD-1 refractory or actually to prevent early resistance?
Both, to be honest with you. And so that's why, again, we would look to you guys to tell us where do you think the best, patient population would be for kind of to first test this out. Now, one thing that's really exciting to me as well is this idea, it was mentioned, actually my specialty is HEMONC, so I'm very familiar with this idea of in lymphoma where we treat the patient, get the PET scan, and if after 3 cycles, and then do something else. And that was one of the presentations. I thought that was very neat. So here we could imagine, for example, based on some of the experience we have, that you treat someone with ipi-nivo, and if they're having not 100% response or maybe less than a successful response, immediately add on or go into our drug. And again, the reason we're excited about it is because whereas there have been 5,000 trials, using various combinations of the checkpoint inhibitors, this is a different mechanism of action.
Jonathan, the FDA is becoming more and more rigorous around multiple dosing, phase 1, multiple cohorts, maximizing efficacy. You went over that relatively quickly. How are you going to fulfill those criteria? You've got a novel drug, and if it's novel, it's going to be tested perhaps more rigorously to make sure you've got the right dose and looking at multiple dosing cohorts. What's your strategy there?
Yeah, so one thing that I think, so our strategy was in the phase 1, we tried multiple different regimens. And we were actually complimented by a bunch of the investigators saying, we think it's great that you're really going to try to find the best dosing regimen rather than just giving it every day. And so I think after actually trying that for a while, we're at a point now where we think we have a good dosing regimen. And then, but you're right, it's probably going to be a lot of tinkering because again, and I don't think this is thought of a lot, there are no small molecule inhibitor IO drugs that are out there. And so we're going to have to figure this out, and it's been a credit to our PIs as well as the Calco itself to take the time to figure this out. So.
Excellent. Tom has stopped taking questions, but I think Chuck has a question. So why don't we go to Chuck?
Just a real quick question. So you mentioned that PTPN inhibition has 2 mechanisms. It affects the killer potency of the T cells, but also renders tumor cells more susceptible to T cell mediated killing. Do you know the mechanism by which tumor cells are rendered more susceptible to T-cell mediated killing?
Yeah, that's through susceptibility to gamma interferon.
But how? I mean, do they upregulate the receptor, downstream signaling pathways?
So one thing is that P2N2 inhibits the JAK-STAT pathway, so that's one of the mechanisms. And also, since you spoke up, I forgot to mention that we're still working on it, but we also have some nice data now in combination with bispecifics, CD3 engagers, as well as ADCs. So again, we think this has a lot of room to combine with a lot of other agents.
Thanks.
Other questions? I'm glad to walk out now that Tom has sat down. Jonathan, can you talk a little about your tox profile? I mean, typically agents that amplify the PD-1 response also amplify the tox. Why do you think your agent is potentially amplifying the efficacy without increasing the tox? Is it because of the tumor intrinsic effect, or is it, what else do you think is happening there?
I think that, so I can't tell you exactly the tox, right? But I can tell you that it's not the same tox. We're not seeing synergistic tox or IO-mediated tox when we add it with PD-1 or by itself. But what I think the answer to your question is that our mechanism of action is different. Remember, checkpoint inhibitors in the real world are there to prevent autoimmunity, right? And a great example is the patients that develop type 1 diabetes on PD-1. I mean, that's like the only thing keeping that patient from diabetes is PD-1, and then they happen to get an anti-PD-1 antibody, right? So I think that has to do with it. So our regulatory mechanism is not an autoimmunity mechanism. I think it's more along the lines of like a modulation mechanism. So a little hand waving, but that's what we think.
And last question for me, was that the BI patient, the kidney cancer patient you showed on the slide there?
So the first one was lung cancer. The second one was kidney cancer, but it's not even our best kidney cancer patient that came from your institution.
You went over that quickly, but I noticed it was a very good response. Excellent. Thank you, Jonathan. Great talk.
Should we back on, David?
What did you say?
Shall we back on?
Yeah, we're back on, and we're still way ahead of schedule.
Well, actually, we're a few minutes
We're
ahead.
2 minutes and 12 seconds ahead. Well,
let's see.
Yes, Minetta has been granted 2+ minutes.
Let's see if I can fill it.
Well, don't feel you need to.
So, hello everybody. Thank you very much for staying. I realize there's not a whole lot of time. I hope you have flights today or tomorrow and get home safely. I wanted to say thank you to the meeting organizers and our session co-chairs for inviting me to present on behalf of Natera. I also want to take this opportunity to thank our past, current, and future collaborators. I learn every day from every conversation that I have with you all, and so does my team, and that's how we're actually going to do better, by working together. So Natera started as a cell-free DNA company focused in non-invasive prenatal testing. So we have a women's health part of the business, we have an organ health part of the business focusing on organ transplant rejection, and then oncology. We have a portfolio of many assays. Here we're focusing on molecular residual disease monitoring. Because we have commercial coverage through Medicare as well as some private payers, we have a commercial volume that enables us to create real-world data, right? We have sequencing data, the lab test results, and we can get clinical information in collaboration with folks, and that gives us a real rich environment to do research in addition to funded studies. We also have the ability to do genomic profiling and additional research services. We have invested a lot in research and development, which from my perspective is not just about assay development, but it's also about clinical studies and evidence generation. We don't want to just put a product out there. We want to be sure we're doing this responsibly and we're all learning together. We have over 125 publications, 30 tumor types that have been studied, and over 20 prospective studies across multiple tumor types. We have a lot of oncologists who are ordering the test, many patients who have been tested. So we're learning a lot, but we need to, again, learn more. We're going to focus on Signatera, which is a tumor-specific, tumor-informed, personalized molecular residual disease test. This requires both tumor tissue and plasma. This involves sequencing of both tumor and normal. to select clonal variants and the highest quality clonal variants possible. So our proprietary algorithms for our exome assay, which is the current product out there, will select the top 16 variants. Again, these are clonal, these are not the drivers subjected to changes in clonal evolution, because we want to look at molecular residual disease. We will custom design an assay based on those 16 variants. By using methylated multiplex PCR, we're able to focus on those 16 variants and follow up with next generation sequencing to sequence really deep, right? So this is targeted and deep focus on those 16 variants. This is a fingerprint. Each test is for a single tumor in a single patient. If a patient has 2 primaries, we have to develop 2 assays for that same patient. That's how specific this is. Once we have that locked down, it's a blood-only test, and we will report this in mean tumor molecules per mil. So it's actually a concentration of tumor molecules, which is different than looking at VAF, for example. We have consistent clinical validation across multiple tumor types. I realize this is small. This is 8 different tumor types looking at recurrence-free or disease-free survival. Green, there's the top line, is ctDNA negative, persistently negative, and the blue or the lower line is ctDNA anytime positive. And you can see how widely separated these curves are, how highly statistically significant the difference in outcomes is favoring those patients who are ctDNA negative. With that early clinical validation, that affords us an opportunity to look at the clinical applications of Signatera, of ctDNA or MRD testing across the patient journey. And because this is developed from the tumor, this is actually applicable across all solid tumors. We're focusing here on renal cell carcinoma, so think about the questions that you have when you face your patients every day and where you might use MRD testing to help you make decisions. Anywhere from the time of diagnosis all the way through the continuum, if a patient unfortunately recurs, looking at treatment response monitoring in the advanced setting. People think about the sensitivity of detection, meaning the sensitivity of detecting a tumor that you know is still in the patient, meaning a tumor you could see or potentially feel. I want you to think about the fact that when we talk about sensitivity of detection, it's really detecting recurrence and using the recurrence as a surrogate for molecular residual disease, which you cannot see in any other way other than through the blood. There are a few studies that I'll share with you for the sake of time. I know I have a couple extra minutes maybe, but I'm not going to go into too much detail. These are all in the public domain. We're happy to send you abstracts, posters, et cetera. This is a study looking real world, so 63 patients, stages 1 to 4 renal cell carcinoma, focusing here on the post-surgical pre-treatment time point, so we call that the landmark or MRD time point, and how that correlates with ultimate recurrence. So looking at recurrence-free survival probability, so these patients all had surgery. If they had stage 4 disease, they had a metastasectomy, they were NED afterwards, and with that single time point, you can see how the ctDNA negative patients did far better than the patients who were ctDNA positive. If we focus in metastatic renal cell carcinoma, this is 92 patients, again, a real-world data cohort. On the left is treatment response monitoring, meaning patients actively on therapy with serial time points, and then in surveillance where patients had completed therapy and were just in the monitoring phase, again with serial time points. And on both sides, you can see that ctDNA negative patients did better. On the left with treatment response monitoring, these were either patients who cleared between before and during therapy, or they were just persistently negative. And then looking at dynamics, this is focusing specifically on patients on TKI. So here this is sunitinib, metastatic renal cell carcinoma, patients where there was a blood draw before starting therapy and then at the end of cycle 1. Baseline ctDNA in this metastatic population was detected in 81% of patients. And you can see the breakdown of those patients who either totally cleared ctDNA, achieved some decrease in ctDNA levels, or had an increase. And these are just absolute numbers. Remember this, we're looking at concentrations because we report Signatera out in mean tumor molecules per mil. And as you can see here, those patients where ctDNA levels increased, after 6 weeks of sunitinib, their outcomes were far worse than those patients who, in the top line there, who had at least a 50% decrease in ctDNA levels. And to be clear, the bottom line, the patients who weren't doing so well, those are patients who either had a decrease in less than 50% or no decrease at all or an increase. So the data I just shared with you is based on our West-based Signatera, so whole exome sequencing of tumor and normal to generate the 16 variant assay. And we realize technology is evolving, number one. And number two, and very importantly, there are tumor types and indications where Signatera West doesn't perform as well as we'd like it to perform. And one can argue that renal cell carcinoma, kidney cancer, is one of them. So Signatera Oncology, we launched Signatera in 2019, and now just in 2025 we've launched a new version. It's Signatera Genome. It's now commercially available. It's also available for testing in clinical trials. We have Medicare coverage for multiple tumor types for both exome and now genome. This covers colorectal cancer, immunotherapy response monitoring across all cancer types, muscle invasive bladder cancer, breast cancer, ovarian cancer, and now lung cancer as well. Many more to come. Hopefully renal cell is in our future, near future. So just a little bit about Signatera Genome. This is data that was presented at the most recent ASCO annual meeting. 392 patients. These are all non-metastatic patients of stages 1, 2, and 3, so the early stage, across breast cancer, non-small cell lung cancer, melanoma, renal cell cancer, as well as colorectal cancer. And what you see here first is that ctDNA positivity after surgery, so single time point, we call that the landmark time point, was associated with significantly worse distant recurrence-free survival compared to ctDNA negativity. So recapitulating what we've seen, right, with Signatera exome. Patients who were ctDNA positive after surgery were those patients who benefited, right, in terms of distant recurrence-free survival. Here, 34% if they received adjuvant therapy. For those patients who were ctDNA negative, they didn't really benefit from adjuvant therapy, suggesting it's such a strong prognostic factor that will identify that population of patients that need additional therapy. And then looking at just distant recurrence-free survival again here, patients who are serially ctDNA negative after completing definitive treatment had almost a 99% 24-month distant recurrence-free survival. This is just the breakdown of patients that was included in that analysis, and you can see the sensitivity and specificity of both the landmark time point, meaning after surgery before starting treatment, and then longitudinally, if you want to focus on the third row, which is renal cell carcinoma. The sensitivity at landmark was 50%. It became 100% with longitudinal testing. Specificity, very importantly, is 100% in both settings. So we were very grateful to see that our sensitivity of detection for recurrence improved. Moving to genome over exome specificity, we lost nothing, and that is critical. Usually you see a little bit of sacrifice in specificity when you get there. People ask us about lead time, meaning when you see a signal in the blood before you're going to see something on imaging. Here we had a gain in lead time of about 3 months, and that may open that window to treat on what we call molecular recurrence, meaning because we know that if ctDNA is detected, a patient is going to recur. If you can capture that patient before they've clinically recurred, that may be the time that we achieve that sort of second chance at cure. We have a product portfolio now for MRD detection. It's both the exome, the genome, and just for your awareness as well, we now have Latitude, which is a tissue-free MRD test. It will launch soon for colorectal cancer, and then in 2026 for multiple cancers. And that's it. If you have questions, happy to answer them. Thank you very much.
Thank you very much. Raise your hand if you have questions for Minetta. Anyone with questions? Yes.
Darren.
Darren again. No, bring it on. We're glad. We like questions, particularly ones we don't have to answer. No.
My question's to you, David. No, I'm just kidding. I have a question about whether you've learned any lessons about shedding of tumor DNA into the blood. Some tumors seem to have more shedding than others, and that seems to, affect the utility for various tumor types, and how RCC compares to other tumors in that way.
Sure, no, that's a great and very important question. So we use ctDNA detection as a surrogate for shedding, right? Because we actually don't have a mechanism to measure active shedding in a patient. For renal cell carcinoma, based on studies that were focused on driver mutations, so not on Signatera, although now we can look at this in our own commercial volumes, RCC is lower in terms of what you would consider shedding in terms of ctDNA detectability or concentrations versus other solid tumor types. And that's one reason why increasing the sensitivity of detection by using a genome-based version of Signatera affords us an increase in sensitivity of detection. There are phenomena that are not just about shedding as well, if you're sort of, aficionados of ctDNA. What we're also learning in the tumor microenvironment, there are actually enzymes that cleave DNA as it's getting secreted. So the ctDNA may be coming out of the tumor, right, through secretion, apoptosis, necrosis, et cetera, but it just may never get into the bloodstream for us to detect it.
Great. One question down front there.
Hey, Manetta, Eric Singer, great to see you. In terms of patients with large tumors and potential tumor heterogeneity or with metastatic disease with kidney in place, do you recommend that we send multiple samples from the primary if it's a large tumor? And if there's multiple sites of disease, do you recommend biopsying metastatic sites, given that the tissue-informed assay is very specific to each of those tumor clones?
So it is a tumor-specific assay, but because the focus is on molecular residual disease and detecting clonal variants, those variants are conserved throughout the tumor despite heterogeneity and in a primary versus a met. And there have been some data we haven't published where we have looked at even the same primary before and after neoadjuvant therapy, with the concerns that that intervening therapy might alter the genomic profile, looking at primary and met as well. And to develop Signatera, so basically we do the same sequencing, run it through the proprietary algorithm, we pretty much get the same test. So my advice for the source of tumor tissue to send to us to generate the assay is the tumor specimen, right, that has the most tumor cellularity, because that's really key for us to be able to, particularly for genome.
Thanks so much.
Thanks for the question.
One last question.
So did you generate data also with papillary, or was it only clear cell? And a bit along the same line as Eric, do we have data in patients that were treated with systemic therapy, and ultimately we use that to assess residual disease in major response with primary in place, for instance?
Yes, so the data I presented is the majority of patients have clear cell, but it includes other non-clear cell subtypes. So that's one. We need more patients though, right? These are small studies, so we need to look at more. So biobanks, prospective studies, we want to work with everybody. So that's one thing. And then the on-treatment phenomenon, so I showed the data with sunitinib, right, in metastatic. And that's 6 weeks of therapy, right? We're looking at early clearance or early reduction in ctDNA could be used, right, as an early response marker. And as we, the FDA, everybody is interested in helping to accelerate drug development, there's huge interest in trying to establish ctDNA as a surrogate endpoint.
All right, we'll save other questions for the end because we are no longer on time. Excellent.
Sorry.
We don't want to shortchange Brian. Brian Liu is going to give, what'd you say? Hasta luego. Have a safe travels home. Brian is from KCRS Life Sciences, and he's going to talk to us about comprehensive molecular testing for solid tumors. I'm assuming one of those is kidney cancer, but we'll have to see. Good.
Yes, it is. Thank you very much. And we're not related, by the way, just letting you know. I'm going to be standing here and just talk about KCRS, and I'll represent KCRS in terms of the platform that they provide. So what is normally that we think of when we talk about a biomarker and a drug association is we usually think of looking at specific targets in the patient. What we at KCRS do is essentially we use the platform, which is a little bit more comprehensive. So we look at whole exome sequencing as well as whole transcriptome sequencing so that we can identify potentially all tumor mutations in that patient. And we find that when we couple our whole exome, whole transcriptome sequencing with real-world data, meaning outcomes data that we can have from clinical outcomes, we can enable our partners to identify associations that they may think that were never possible before. And I will illustrate some of that today. So for example, when we take a look at solid tumors, we profile essentially the whole exome sequencing, the whole transcription sequencing, as well as a battery of IHCs. We also have a liquid profile, which is our liquid biopsy. And in that, basically, you can see that we also have a whole exome sequencing on the liquid biopsy, whole transcriptome sequencing on a liquid biopsy. And because we also sequence the Buffy code, we can identify incidental germline as well as to rule out potential false positive that is due to clonal hematopoiesis. And those are important. So I'll give you one example of a tissue report as shown here. In this tissue report, you'll see on the top line, essentially, that this patient has a VHL alteration. You can see a drug, belzutifan, which is associated with the VHL association. Because we also sequence in this patient or the tissue more than just a target for looking at VHL alteration, we found that there are other genes that this patient also have. In this case, an ATM alteration, a BAP1 alteration. And we also look at other genes that I don't know if you can read on the screen, but essentially we found that we looked at PD-L1, MET alterations, and others that are not determined on this particular tissue. When we take a look at a liquid biopsy, same approach. On the top line, we found that basically this patient, a separate one from the tissue, that we have another VHL alteration. This is found in the germline, and we could pick that up because we sequenced the Buffy code. We also know, basically, from the plasma, which is essentially shed circulating nucleic acids from the somatic tissue, that this patient also have an MSH2 alteration from the tissue. If you go down the list at the bottom, when you take a look at clonal hematopoiesis, there's no false positive from the CH in this particular assay. Why is this even important? Because we want to make sure that we're ruling out false positive, which may play a role in some of these therapies. Now, one thing is that basically we have our database that is a clinical outcome, and you can see on this clinical database that we can merge with our genetic molecular database. And this provides us with associations and allow partners to really ask questions about what they want to find. So one example of it, I'm not going to go through that, is looking at this HIF transcription factor in over 4,000 renal cancer patients. And you can see in this slide here that basically our question really was, are there association with particular genes with high expression of HIF transcription factor? Well, I'm not going to go through this slide because all of you are aware of what HIF transcription factor can do, but just to remind you that when you have a VHL alterations, there's an accumulation of HIF2 that can turn on genes, leading to angiogenesis, proliferations, and so forth. The data from the poster shows the following. When you look at the highest quartile of HIF2, which is on the left, the top bar, you can see that there's more alterations in those patients with VHL than the lowest HIF2 quartile. They tend to be less. You can see association between the highest HIF2 alpha and other genes as well. So there's different drivers, different associations, some of which are positive correlations, some of which may be negative correlation. And if you take a look at outcomes data on the bottom, where you take a look at the Kaplan-Meier curves, the overall survival, you can see that those who are expressing the highest level of HIF2 alpha tend to have a highest or better overall survival than those with the lowest HIF2 expression. And the same thing in terms of looking at time on treatment with cabozantinib. If you were to take a look at that, you can see that the patients that has the highest expression of HIF2 alpha tends to be on longer cabozantinib than essentially those with a lowest quartile of HIF2 expression. And you can take a look at HIF1, beta, and alpha, and you can see that there's no discrimination between cabozantinib on either one of those transcription factors. So hopefully what I want to illustrate to you today is that when you do a molecular comprehensive profile and you merge it with the clinical data, we can provide and partner with those who are interested into generating association outcomes and potentially help you identify markers as well as therapeutic targets. And that's really all I have to say. So thank you for your time.
So questions for Brian? We have some time.
We have 3 minutes. Could I ask a question for Vanessa? Vanessa, could I ask you where you think ctDNA is going to be in kidney cancer 2 years from now?
For me?
For Vanessa, if that's okay. I'm going to ask that.
2 years from now?
In 2 years from now. So at the moment, the summary of I think where we are at the moment is even with your most recent test, although there are only 25 patients, the sensitivity was 50%, which is not great. That suggests that it's probably not going to change the world as it stands. Is there a cohort out there of 1,000 patients or 100 patients which is more accurate? Are you going to generate that data? Or as you said, or as you alluded to, there are other areas where this is probably more promising and something that you're just going to leave behind.
It felt like a leading question, but go ahead, Minetta, try to answer it.
I will comment back.
Yes.
We will not leave renal cell carcinoma behind. There's really no tumor that I think we want to leave behind. Okay, we are committed across the board, so that's one thing. The 50% sensitivity was single time point after surgery before treatment, that landmark time point, but our sensitivity improves significantly. It's 100% with longitudinal testing. Okay, so
Can I push
like
you on that a tiny bit, if I may, just because eventually everyone's going to become CTA positive. So so longitudinal testing, the 20-minute, multiple liver material. So longitudinal testing has got lots of flaws in it.
It does, it does. But just to be clear, just from the, I get it though. Single time, it also depends on when that time point is selected, which is why we use the landmark, because that'll be consistent for everyone. Our genome product is out there. It's a little bit better than exome. We are working on another version to account for tumor types where including introns and those additional high-quality variants across the genome have given us some but not enough. So once you do whole genome sequencing, it gives you an opportunity to look at other variants. And I mean, others are doing this, but I think that the magic is in our informatics team. So we're actively looking into that. So we're not going to just stop here. So that's the product. And then in terms of studies, yes, this was initial very small numbers because we're just getting started. We have several biobanks that we're working on now, and as I mentioned earlier, we're always looking for more. So we will be able to return the sequencing data back to the collaborators. We do want to work with people. I can't stress that enough. And we're also willing to invest in prospective collections because to collect specimens well that are clinically annotated, again, will get us the answer faster. In 2 years, I hope we start some interventional trials.
Thank you. Tony, it's your last question.
You have 8 seconds.
Yes.
How much?
8.
5
No,
now.
5. I worry that when you have with the sensitivity that of 50% that is low, by the way, our experience have shown way less, even in stage 3B and stage 4, is that we're going to have patients with negative scan, positive ctDNA, and they would want to ask for pembrolizumab, an adjuvant, and that is going to be something very hard. So I would love to push you harder to get not just whole genome, all intronic variant, etc., so that we're more sure. We don't have that problem in bladder, but when we had it, Genentech designed a study, InVigor 011. So that is led by Dr. McDermott, not Dr. Powles. So that's going to be a problem, giving pembrolizumab based on ctDNA positive.
That feels more like a statement than a question. You don't have to answer that if you don't want to.
Well, I just want to say that we don't know that it's going to be a problem. We just haven't proven that you need to treat with ctDNA positivity in the absence of clinical recurrence.
You are not in front of the patient. I'm sorry, but
I was in front of the patient.
no,
just
no.
2 years
So you want to tell me a ctDNA positive patient that want pembrolizumab, you want to treat based on ctDNA positivity?
So there are clinical trials, as you know,
But
right? where
but
we're-
today in practice, when I order the
Not in
test,
practice.
but then why would I order it? Why would I order it if the scans are negative?
So right now, for the why people
Take the
tell
box
us
a-
that they're
take the
ordering
box
it
away from them.
commercially, maybe I need to buy you a beer. But why people are ordering it commercially is to help guide surveillance in those patients. They're not going to treat. We don't have data, right, to start treating on molecular-only recurrence, but certainly the ability to continue to scan patients and have higher vigilance to make sure that you're finding.
Okay, let's pause it there. We can continue this conversation on the side. We have our CEO is waiting to wrap up the meeting. So why don't we thank all of our speakers.